Hi everyone, welcome back to the channel. It's been quite some time since I've put out a video. I've been quite busy with work and with a recent move that my lab is undertaking. We are shifting from Michigan State University across the way to Ann Arbor at University of Michigan. So we're pretty excited about the move. The lab space is, quite a big upgrade. Super cool. We're super excited about it. But it has made me very busy. And so, this YouTube channel is one of the first things that's going to fall to the wayside when I get busy IRL. But I wanted to take a second and kind of go over a letter that I wrote to a YouTube creationist that some of you might be familiar with. His name's Donnie, and he runs the Standing for Truth channel. And for those of you that aren't aware, this is the Standing for Truth channel. I recommend you check it out if you're interested in like the history, like Bible history, Bible translations. There's some cool debates that they do, like soteriology debates. This is like the doctrine of salvation. They have some neat things regarding some upcoming debates on like Protestantism and Catholicism that I'm looking forward to. So if that's the sort of thing that you're interested in, then I definitely recommend checking the channel out. They all Donnie is also well known, obviously, for being a staunch creationist and for hosting creation and evolution debates. And these I tend to find a bit more frustrating, especially the evolution on trial ones, the evolution challenge debate series with Ken Hovind, because I find the man absolutely insufferable, such that it's very difficult for me to watch him speak for more than 5 or 6 minutes. Because I just feel like my ears are going to start bleeding. So with that being said, he actually put out a video. He, as in Donnie, has a video on Kent Hovind's channel in which he presented some of his recent work on endogenous retroviruses. So if you don't know, Donnie wrote a book, sort of debunking the idea of endogenous retroviruses as evidence for evolution. And, it's an hour long presentation. If you're interested, check it out. But after watching it, I thought, you know what, I'm going to take a moment and kind of summarize all of my thoughts and write Donnie an email. And as you can see, this email is quite long, with even some figures in it. So, it was an extensive email that took me the afternoon to put together. And I sent it on August 4th. It is now August 25th. Donnie has not responded, and I don't say that in a way that's like, oh, Donnie chickened out and didn't want to respond. No, it's like, it's a long email, for one, and, he doesn't owe me a response. I'm sure he's busy. Like I said, he's got tons of debates that he does. Um, and so that's perfectly fine. This is not a pot shot at Donnie for not responding. Um, Instead, what I'm hoping that this shows is for those of you that have watched some of his debates, that have watched him speak, or that are interested in these topics, I hope to use this as more of like an educational opportunity because I try to delve into some of the nuance of the discussion that I think is missed oftentimes in these YouTube back and forths. So what we're going to do today, again, very different from what I've done previously, is just kind of read through this letter. This may be boring, it may be terrible YouTube content, but I think some of the points are interesting, and I really tried to put a lot of thought and effort into this email. And so if Donnie, literally never responds to it, I feel like that's a lot of work that I put into, and nobody else will ever see it, assuming he even read it. Which, again, he has, no obligation to read and respond to this letter, so that's not my point here. Okay, so just to kind of jump on into it, on the subject line, I put thoughts from an evolutionary biologist. Again, I try to hearken on this part, not as a brag, but as a like, let's see if I can get him to engage with me by pointing out my credentials. So, I start off hi Donnie, my name's Zach. I'm a postdoctoral researcher. I'm an evolutionary biologist. I specialize in popgen, phylogenetics, genome evolution. I tell him, where I got my PhD from. Um, and then I list my Google Scholar here so he can check it out. He can see the papers that I've written, that are peer reviewed, part of the scientific literature. I also link my GitHub here so that, like, if he's interested in some of the modeling stuff that I talk about later on in the letter, he can go to any of this GitHub stuff. He can check out my code, etc., etc. So again, and I try to preface this with, like, this is not some weird brag. I'm just trying to establish a sense of credibility, right? And then I, just introduce, stumbled on your channel a couple of months ago, watched some of the evolution debates, blah, blah, blah. None of that's really interesting for us here. Okay, so beginning, be that as it may, from your moderating and the few videos I've seen you promote your ERV book, you seem reasonable. He seems like a reasonable guy. I recently watched a video by Dapper Dino, Creationist, Dr. Dan Cardinal, as well as from Gutsy Gibbon, who all argue that Donnie's actually not a reasonable guy, and they give some evidence for this. Again, I don't know the guy, never had a conversation with him, and he seems to present himself, at least in the debates that I have personally watched, as a reasonable human being. So I'm gonna pretend that he is. If he's not, then fine. I'm gonna give him the benefit of the doubt here. I also lead with stating that I'm not a professional debater in any sense. I've never had a formal debate. That's, like, not my job title. So what I thought would be the best way to sort of summarize my thoughts from this video, which again, if you're interested in detail, you should watch maybe prior to watching this one. The best way for me to sort of engage with those arguments is to write a letter, right? So I made a list of the points that you made that I found misleading or incorrect, and while it's in no means exhaustive, I think each collectively challenges your perspective on what genomes are and how they function. So starting off, the first topic that I wanted to get into was nearly neutral evolution. And from here, I'm going to, like, largely read what I've written, and I'll just elaborate on points as I go along. So you reference Kimura Noda's work several times in your talk, which I appreciate. They are too often forgotten despite being giants in the field. However, it seems you've misunderstood the implications of their work. Prior to Lewontin and Hubby's work on natural levels of heterozygosity in the 1960s, it was believed that all standing variation was maintained by natural selection, i.e. balancing selection. However, the levels of genetic variation that was later discovered far exceeded what could be maintained by selection. The selective cost, quote unquote, would be too great. See Haldane, 1957, for the mathematical derivations for this. This is somewhat similar to the genetic entropy argument. However, this issue was solved by population geneticist Moto Kimura and independently by biochemists Jack King and Thomas Jukes in 68 and 69, respectively. Kimura, arguing from mathematics, demonstrated that the levels of standing variation scaled with population size across the biological world. And this is a finding that has held up surprisingly well into the genomic era. See Ellegren and Gaultier, 2016, for a review. I've done previous videos where I have looked at this scaling pattern across the tree of life. And you can check those out if you're interested. Further, he showed that the rate of substitution in hemoglobin across mammals scaled linearly with the proposed time of divergence. These two facts, he contended, were consistent with the prediction that most new mutants were selectively neutral, and that the rate of substitution was determined by the population size, i.e. drift. King and Jukes took a slightly different approach from biochemistry, while leaning on Kimura's mathematical intuition. And they point out, for example, that the hypermutating regions of cytochrome C in the mitochondria, that the number of changes per site observed is predictable by a Poisson distribution. And like wildly, this includes not just synonymous site mutations, but also amino acid residues. That these follow such a distribution demonstrates that there is no constraint on which amino acid is substituted, regardless of whether they are neutral, basic, acidic, hydrophilic, or hydrophobic. You stated in your presentation that scientists did not predict 20,000 genes, as if this was greater than the number that was thought. The opposite is true. Many biochemists thought humans had hundreds of thousands of genes, but King and Jukes knew better. Again, in their 69 paper, they noted that if the recessive lethal mutation rate is 10 to the negative 6 per locus per generation, which had been estimated empirically at the time, then the number of functional genes in the human genome could not exceed 40,000. Thus, in 1969, before we had a human genome, I should add, we had an upper limit of the number of coding genes that was mathematically derived following intuitions from neutral evolution that was vindicated decades later. Okay, so now with the important background out of the way, your specific claim. Due to the entire genome being functional, all mutations are deleterious, that deleterious mutations accumulate and cannot be removed by natural selection due to these mutations being nearly neutral, which eventually leads to extinction. However, this misunderstands fitness and nearly neutral theory. If the inequality 1 is greater or equal to 4 n sub e of s is satisfied, where n sub e is the effective population size and s is the selection coefficient, then the mutation, irrespective of s, behaves neutrally. This means that the fitness of the population does not decrease, because the impact of the mutation is indistinguishable from the neutral variance. Of course, if we assume these effects are additive, an assumption that may or may not be true, though I will grant that the field of genetic load assumes that it's true, these mutations can theoretically lead to a mutational load, but the severity of this load is determined importantly by the population size and the recombination rate. If you're interested in why the recombination rate is important, just Google Mueller's ratchet. Okay. And so to go forward with this kind of line of reasoning, it's just been verbal so far, I propose to use some simulations to shed light on this intuition. And if you're curious, he had done a video previously, let's see, in which they were looking at genetic entropy. Where is it? Evolution's evil. God, they do so many on here. This one. Is genetic entropy a legitimate problem for evolution? With T-Rock arguing in the affirmative and Luca Medugno arguing in the negative. So check that out if you're interested in the whole genetic entropy debate that they have. Okay, so, and I actually ran these simulations in response to that debate to try to demonstrate that the intuition of genetic entropy is wrong. So let's get into it. So in the figures below, I simulated a population of 10,000 diploid individuals using SLIM. SLIM is a very popular forward in time simulation software that is used by thousands of researchers around the world, each with a 1 megabase genome, a carrying capacity of 10,000. What that means, a carrying capacity is the number of individuals that the environment can sustain, right? So like if you go beyond that, the environment cannot sustain you because you've depleted the resources of the environment, right? And a mutation rate and recombination rate of 10 to the negative 7. Of these mutations, their distribution of fitness effects were 50% deleterious with a selection coefficient of negative 0.002, 45% neutral, selection is 0, and 5% beneficial, with an S of positive 0.002. So as you can see, this is a distribution of fitness effects that is strongly leaning to the left. Okay, it's strongly left skewed, heavily relying on the majority of the mutations that appear are deleterious or have no fitness benefit, right? These simulations were run for 8,000 generations, and as you can see from the figures below, despite the order of magnitude preference for deleterious mutations, an environmental capacity of only 10,000, the population was able to persist and indeed grow to 13,000 individuals. Okay, so here's figure 1. And on the x-axis is generations, on the y-axis is population size. I've put a black line for what k was set to. Again, the carrying capacity of the population. Initially, at generation 0, you see the deleterious mutations are like bombarding the population, making it dip below that environmental capacity, right? Because again, most of the mutations that are occurring are bad. They're deleterious. And so the population starts to dip, but then at around 2,000 generations, you notice it picks up. And then it just starts to grow and grow and grow and far exceed the carrying capacity. What we're seeing here is that those beneficial mutations, though they occur very, very slightly and only occasionally, are able to not only persist and to conquer and cancel out the deleterious ones, but they are able to increase the overall fitness of the population such that it can exceed the carrying capacity. Again, if genetic entropy were true, this should not be able to happen, right? That this process here should never be able to happen. And yet we can do a simple simulation and show it can. In figure 2, we actually see natural selection at work. And what figure 2 is showing is the ratio of neutral to deleterious mutations. That's on the y-axis. Again, the x-axis is just generations. Under a scenario of mutational load, selection should be incapable of removing deleterious mutations over time, and this ratio should approach 1 to 1. Actually, it should approach some number less than 1 to 1 because deleterious mutations are 5% more likely to occur. So it actually should be below this line. So this is just like an error on my part to say that they should approach 1 to 1. It should actually be slightly below 1 to 1. However, despite the preference for deleterious mutations, selection can remove them. And so we get a ratio that is above 1 to 1, right? And so what this is basically showing us is that there are more neutral mutations than there are deleterious ones. If mutational load were true, right, in all settings, right, and that's important, and I'll get into that in a moment, then it should always be the case that the number of neutral mutations should equal the number of deleterious ones, because the deleterious mutations are behaving as if they're neutral in all settings, according to like the concept of genetic entropy. But again, a very simple mathematical simulation can show us that that's just not the case, and that natural selection can purge deleterious mutations, even if they are the dominant mutation in the distribution of effects. Okay, so importantly, mutational load can be mathematically predicted in the absence of simulation. We don't even have to simulate this, we can mathematically predict it as the cumulative effect of mildly deleterious mutations relative to the recombination rate and the population size. So, for example, I could lower the population size to 100 individuals, keeping everything else constant, the population will collapse due to selection being inefficient, i.e., mutational load. Alternatively, I could set the recombination rate to 0, which similarly reduces selection's efficiency, and it's reducing the effect of population size is like literally what it's doing, and can collapse a population even at larger population sizes. So very important to the entire conversation here, and that was completely missed in Donnie's presentation, and in honestly any presentation on genetic entropy, is that nearly neutral evolution is context dependent. It's dependent on what the population size is, what the selection coefficient is, what the distribution of fitness effects are, and what's the recombination rate, right? All of that is incredibly important to determining whether you are in a neutral, nearly neutral, or genetic load setting. And again, like these numbers that I picked, the S are relatively high. And given that the population size is only 10,000, this is like human population size, long-term human effective population size. A lot of natural populations are much larger than this, right? So like selection is going to be even more efficient in those than it is in the simulations I ran. So it's just preposterous. It's just preposterous to think that selection can't weed these out. All right, so finally you stated that evolutionary theory requires genomes of junk. This misunderstands King and Jukes' point. It is not that the theory requires genomes of junk, it's that reality requires it. If our empirical estimates are correct regarding the mutation rate, then the genome could not persist if wholly functional. For organisms with short generation times, we should see consistent population size declines even within a human lifetime, especially given that the amount of standing variation in these populations. However, we do not observe this. Bacterial populations are not vanishing around us, right? Like that's just not what we observe at all, despite the fact that they're making this claim that all mutations are bad and that all populations are winding to extinction because of genetic entropy. We just don't see that. It's just not observed in reality. So TLDR, nearly neutral theory does not predict genetic entropy. That's not what it predicts at all. It predicts that the fate of an allele is not due solely to the selection coefficient, but its interaction with the population size. A mutation that satisfies this inequality is selectively neutral and has no effect on fitness because the fitness cost, right, the negative fitness cost, does not exceed the neutral variance in offspring number or mean lifespan. Okay, it looks just like a completely neutral mutation. It's not passing the neutral variance. Therefore, it does not have an effect on fitness. It is only by assuming that all of these independent mutations have some cumulative effect on fitness. And again, this may or may not be true, right? This is an assumption of genetic load literature that may or may not be true in nature, especially considering that that recombination exists. Okay, so moving on to point 2, junk DNA. This is a favorite topic of mine. I've made an entire video on this very thing. It's a talking point of Creationist that, and Donnie has said in other videos that junk DNA is not a thing, and that it is a prediction of creationism that, or at least his brand of creationism, that the human genome must be wholly functional, right? Okay, and again, he said that in the presentation, genomes are 100% functional. This is not supported by genomic research, and I'd like to discuss why. First, let's consider how you're defining function. In molecular biology, it is often assumed that biochemical activity is a function. This is because molecular biologists are descriptive in their work. X does Y does Z. This is the causal definition of function. But that's not how you're using function. You're using the selected effect definition, which states that X resulting in Y does so because it is necessary to produce Y, or that failing to produce Y contributes to a fitness decline. It's very important to understand why these are different, and then I give a bunch of citations discussing these differences. Now, let's consider why the causal definition of function is inappropriate. What is the function of a shoe? Well, one thing a shoe does is bind gum from a sidewalk. That is an effect that is caused by a shoe. Thus, under the causal definition, a shoe's function is gum binding. This is obviously ridiculous. A shoe functions to protect your feet. A shoe could become so damaged that it no longer protects your feet, but still manages to bind gum. You'd say that shoe does not have a function anymore, despite carrying out the activity of binding gum. I should state that this example is from Dan Grauer's 2013 paper. Okay, so in a biochemical setting, the same is true. That the genome shows some biochemical activity does not alone demonstrate function. Palazzo and Keiju in 2022 recently discussed this from a biochemistry and cell biology perspective. First, they note that cells are sloppy. Enzymes often engage in non-canonical activity, including binding with proteins and metabolites that cause the enzyme to cease to function properly. The sea of possible interactions of enzymes and metabolites is called the interactome, and it's exceptionally messy and prone to error. Any student of organic chemistry understands this. For every reaction, there is a non-canonical product. The probability ratio of this product is given by this equation, where E sub n is the energy to produce one product, K is Boltzmann's constant, and T is temperature. Importantly, the only way to only get one product from this equation is if this energy differential is infinite. That's the only way. That's the only way you could just get one product, which means you will always get, at some probability, the product that you didn't want, the product that that particular gene didn't actually code for, or that that particular protein wasn't actually meant to create. You will get a non-canonical product. Studies have found that the rate of protein misincorporation, i.e. non-canonical products, can be as high as 10 to the negative 3, meaning that most produced proteins have some sort of error relative to the DNA sequence specificity. Transcription is even worse. It's widely observed phenomenon that transcription can be activated by random pieces of DNA across the genome due to transcription factor binding sites being relatively short compared to the total genomic length. Indeed, in the field of transcriptomics, the largest hurdle to overcome is transcriptional noise, in which I've had conversations with researchers that have stated that this transcriptional noise can be upwards of 70% of the transcripts in their gene expression studies. Lots of transcriptional noise, lots of just messy mRNA transcripts floating around in any given cell at any given time. Transposable elements contribute heavily to this, as do other forms of repetitive non-coding DNA. Further, transcripts themselves, that is, mRNA transcripts themselves, can become incorporated into the genome thanks to reverse transcriptase action from transposable elements. We find these litters across the genome. These are post-processed mRNA transcripts inserted into the genome lacking promoters, transcription factor binding sites, and introns. Right? They're post-processed mRNA. You've already spliced out the introns. They're just the exon, just what's supposed to be the coding region. They should be on their way out to the ribosome, but they get snagged up by a reverse transcriptase protein and just plopped back into the genome. They don't want to be there, right? They're dead on arrival. That's not where they're meant to be. Our genome has tons of these. Our genome is also littered with pneumites. These are nuclear mitochondrial elements that arrive from a similar process. Post-processed mRNAs from the mitochondria that accidentally enter the nucleus and become incorporated due to the random reverse transcriptase proteins. Okay, these again are dead on arrival. The nuclear genome cannot produce mitochondria-specific genes. When you look at these pneumites, when you actually sequence these particular pneumites, they have frame shift mutations. They're littered with stop codons. They are not producing functional proteins. There's just no way when you look at those sequences can you find any evidence that they're, one, sequence constrained, or two, actually capable of producing functional proteins. Insertional processes also generate clear evidence of dead on arrival genes. For example, lines, these are prolific elements in eukaryotic genomes, are infamous for producing poor products. This is because oftentimes reverse transcription will be activated at the 3' end of the flanking DNA. Okay? That's not where you want transcription to begin, and which means that it very often will fail to reach the 5' terminus. This truncates the sequence and leads to the insertion of just a fragment. All right? And indeed, some 50% of the human genome is made up of these transcriptional, or these transposable element fragments. All right, all of this is to say that biochemical activity alone is insufficient to demonstrate function, right? All of that, what I just said, was different types of biochemical activity, right? But nobody would say that those things were functional, right? That the action of inserting a post-processed mRNA into the genome is a functional act, right? Because these cells, as I said, are sloppy and are highly error-prone. Okay, so what then is an appropriate way to identify function? First, we need a model with some testable predictions. We can construct such a model using the intuition provided from Kimura and others that we talked about above. Let us imagine a model of no selection, in which the amount of standing nucleotide diversity is merely a reflection of mutational and drift processes. When a region matches our model, we can designate it as under no selective constraint, i.e. it is not functional. To assume that a region can match expectations without selection and yet still be functional assumes that 1, there is never any deleterious mutation, which is absurd, or 2, no mutations ever occur there, which is also absurd. We could broaden our model to size variance, binding affinity, and any other component of the 3D structure of DNA as well, pretty simply, but for brevity, I'll only introduce a model of nucleotide constraint. The amount of expected standing diversity is what we call Watterson's theta, and that is pi equals 4 n sub b of mu of L, where L is the length of the genome, mu is the mutation, and mu is the mutation. Importantly, this model is standing diversity at equilibrium, which means that regions under selection should depress pi, or genetic diversity, relative to 1, the strength of selection, and 2, the recombination rate. Amazingly, we see these patterns across eukaryotic genomes. And I give a specific example here below from Nam et al. 2017, a study of primate genetic diversity showed that pi increased logarithmically with distance from known genes, eventually reaching an asymptote that is expected given our model. The equilibrium value of pi was greater for chimps and orangutans than for humans, primates with known larger long-term n sub e than humans, exactly as our model predicts. So here's the figure that I shared. You can note the dip in diversity the closer to a known gene and the subsequent exponential increase with distance, followed by a flattening or an asymptote. And that this flattening of pi reflects the equilibrium value of 4 n sub e of mu as predicted by neutral theory. So the x-axis is distance to the nearest gene, where 0 is the nearest gene, and then you're getting further and further away. And then y is genetic diversity. Okay, and so in each of these you can see this dip, this much lower level of genetic diversity, which then rises and flattens, right? You see this pattern in all 4 species. Furthermore, our model would predict that if the majority of the genome was not under sequence constraint, then the levels of standing diversity should scale with population size. The reason for this is that mu only varies by 1 to 3 orders of magnitude, whereas n sub e varies by dozens, right? And should therefore drive patterns of pi. And indeed, this is exactly what we see. And here's a figure from Elegren and Gaultier showing this. 2 proxies for population size, that's fecundity on the x, propagule size on the y, scale exceptionally well with genetic diversity, which is the colored points here. This is not predicted from genome-wide functionality, but rather from neutral theory. All right, ERVs, which your presentation was dedicated to, also fit this pattern. Most human ERVs do not contain the full repertoire of proviral genes. I think it's like 80 to 90% of them lack the actual genes, but instead exist as just solo LTRs. There is some evidence of purifying selection acting on the POL, GAG, and ENV genes. The mutation rates in the LTRs match the average genome-wide mutation rate, and I give a few studies showing that. Furthermore, extensive studies on the gypsy element, which is a type of ERV, in Drosophila, fruit flies, organisms that we have studied in labs extensively, have found evidence that these elements are transmitted horizontally and can be incorporated from the female somatic tissue by expressing itself and becoming transmitted to the oocyte. This is a very interesting finding, which indicates that these ERVs are still behaving like viruses and could explain why purifying selection is maintaining sequence identity. The purifying selection is acting on the virus itself, not on the host. Indeed, following this, there's evidence of RNA suppression of the gypsy element within Drosophila ovaries, which again would indicate that this element is a virus. I bring up the gypsy element because it's ancient, and it's prevalent in many species, including humans. I studied amphipod crustaceans in graduate school, and we found gypsy elements in amphipod crustaceans as well. All right, so finally, ERVs bear the hallmark of viral insertion, target site duplication, which occurs following the cleaving of a target site, insertion of the viral element, and then DNA repair of the cleaved target site, right? So what you end up seeing is identical target sites on either side of the viral element. And this is just due to the insertional process, right? You see this duplication is seen in many transposable elements as well that are known to jump around in the genome. And so if herbs were created already in place, as Donnie supposes in his presentation, there's no reason to expect target site duplications flanking viral regions. There's just no reason to expect them to be there. All right. Finally, an important consideration rarely discussed in respect to human genome functionality is the incredible variation in genome size across organisms. And indeed, even between two humans, the genome can vary in size by several megabases. This is a thing that, like, I've never heard a creationist address, but genome sizes aren't constant, right? Some humans have more genetic material than others. There's just variation in how big a genome is. And all of that variation comes about because there's variation in the number of repetitive elements that you have, and that those can expand and contract willy-nilly, like there's no selective constraint on them. And so by that very fact, any two individuals in a population can have different genome sizes. As I stated previously, I studied genome evolution of amphipod crustaceans. Congeners, these are species within the same genus, had genome sizes that varied from 2 to 14 gigabases. 2 being 1 gigabase smaller than the human genome, 14 being several times larger than the human genome. And that these differences were entirely explained by the proliferation or purging of repetitive elements, especially lines. If these elements were fully functional, there's no reason to expect, one, that they can be purged, right, that natural selection would be acting to remove them, or two, that congeners should have such dramatic differences in their genome sizes. And yet we've found that across biological life, there is a clear scaling of genome size with intergenic regions, including introns and mobile elements, and that importantly, this scaling is independent of complexity. Some of the largest genomes are unicellular eukaryotes. If we were to prescribe 100% functionality to the human genome, we need to explain why it is that Amoeba dubia has a genome 100 times the size of ours and yet consists of just a single cell. Okay, TLDR, junk DNA is supported by multiple lines of evidence, and the majority of the diversity in the human genome fits a model that does not include selection. Herbs are no exception, with standing levels of diversity comparable to the mutation rate. Finally, genome sizes across the tree of life vary by orders of magnitude and yet do not scale with complexity, demonstrating there cannot be a one-to-one genome size to functionality relationship. All right, the last chunk, the last point that I wanted to address is genetic similarity is just common design. This is a very common talking point that you see from creationists all the time, that two organisms have the same DNA, not because they share an ancestor, but because they were made the same. So I start by stating that this is a curious argument that I suspect you don't entirely believe. I'm sure you accept the findings of ancestry services and paternity tests, which rely on genetic similarity reflecting ancestry, right? It's an observational fact that more related individuals share more unique mutations in common. We see this. This is observed. And it's indeed this fact that I'd like to focus on. Okay, so under a model of common design, genetic similarity should scale perfectly with phenotypic similarity. This is especially true if we assume 100% genome functionality. And for a test of this intuition, consider the following figure. I was inspired to do this figure from some of the debates that I've seen Kent Hovind give, where he's got the cartoons of 3 dogs and the banana, and he says, a 4-year-old can pick out, which one's not like the other, right? Like, you see that all the time. And every single time he shows it, I think, well, those are obvious examples, but most life is actually a bit more complicated than that, right? And that you could, like, a 4-year-old would actually get the relationships very wrong, right? And that prior to genetic evidence, we got a lot of these relationships wrong. So I wanted to give him a bit more of an adult example. So These are the 5 critters that I settled on, and I asked Donnie to predict which of these, like, what's the genetic similarity between these 5 critters. The first one, A, is a gooseneck barnacle. B is a gammarid amphipod. This is a marine amphipod. C is a terrestrial isopod. You can call this a roly-poly. D is a silverfish, a terrestrial hexapod. And E is a Penaeid shrimp. These are the tasty shrimp that we enjoy in Alfredo pasta. So again, can you predict which of these should be the most genetically similar? So under model of common design, I would predict, and again, I don't know if Donnie would make the same prediction, but this is what I would think, that E and B should be the most similar. E and B, because they both kind of look shrimpy, right? They're the most shrimpy looking. D as slightly more distant, because it's like, doesn't have quite as many legs, but it does still look kind of shrimpy, right? Like, silverfish kind of have a shrimpy vibe to them. They got the long antennae, the legs, the narrow body, with clear segmentations, they're kind of shrimpy. C as more distant. This is less shrimpy, but has the segmentation, has a lot of legs, has the antennae, right? Like it's, obviously different from these 3. And then finally, A is the most distant, because, like, look at this thing. Like, there's no universe in which you look at this critter and say that it is closer to any of these 4 than any of these 4 are to each other, right? Like there's just no universe in which you say that under the idea that common design explains genetic similarity, right? However, of course, this is false. Our relationship we just proposed is wrong. Instead, the real relationship is CB, which is isopod, amphipod. These are paracarids, they're sister taxa. E as more distant, the Penaeid shrimp. So these 2 as sister, followed by the Penaeid shrimp. These 3 collectively are malacca-stalking crustaceans. And then A, C, B, and E, so these 2, this one, followed by the barnacle, because these 4 are allocrustaceans, with the most distant being the hexapod. And then here is the phylogenetic tree, and then I just like plucked the same pictures out and then gave their relationships here, showing the phylogenetic affinities. If the genome did not reflect ancestry, but only common design, how could this possibly be true? Gooseneck barnacles are obviously more phenotypically distant than all the other critters I just showed, and yet they nestle within crustaceans. We could perform this exercise across the tree of life. For example, McKenna et al. 2019, examining the phylogenetics of beetles using whole genome data, found that carabids and polyphagid beetles diverged around 310 million years ago. Compare this to Upham et al. 2019 on mammalian diversity, which found humans diverged from mice only 170 million years ago. This indicates that 2 coleopteran beetles, are nearly twice as genetically divergent as humans are to mice. These confusing patterns are immediately removed if we consider that genetic similarity reflects ancestry. Right? It reflects ancestry in conjunction with what we've discussed above. 1, most of the genome is not functional. Therefore, most substitutions are neutral, and that the genetic divergence between any 2 species should reflect time since their last common ancestor. Note that this says nothing about phenotypic similarity. It doesn't say that any of these 2 should be the most phenotypically similar. It's while genetic and phenotypic similarity tend to be related, they don't need to always be. Unlike the claim from common design and presuming 100% genome functionality. Right. And again, this is because the bulk of the changes between any 2 species may be in neutral regions of the genome. Right? That the divergence may be in regions that aren't affecting phenotype. So you can be incredibly diverged from one another and look very, very similar, Or you can be very shallowly diverged from one another and look very different. Just depends on where that divergence lies, right? And that's a prediction of evolution that cannot be explained by common design. Especially common design in conjunction with 100% genome functionality. All right, finally, we can consider shared mutations as reflecting ancestry. You stated in your presentation that mutational hotspots make this impossible, but it's actually quite trivial to account for this. In phylogenetics, it's necessary to regress estimated divergence by two substitution models. One is called p-distance, which does not account for back mutations or convergent mutations, and a more complicated model, such as the general time reversible, or GTR, which permits rates of back mutation and independent rates for transitions and transversions. This seems like a bit of a side. One of the things that Donnie stated in his presentation was that we can't use shared mutations as evidence because there are mutational hotspots in the genome that rapidly mutate, and so what we're actually comparing are convergent mutations, not shared ancestry. That was the point he was trying to make, so that's what I'm addressing here. The regression that I'm talking about is a saturation plot in which we plot a line of expected divergence against what we observe. If the observed falls below the expected, which occurs at very deep divergences generally, then we have what we call saturation, right? Where we have as many back mutations as we do like mutations reflecting ancestry, and we can't tell them apart anymore. This means that either one, we can incorporate the more deeply divergent taxa, or we must prune the data set to remove loci with evidence of saturation. For older data sets, what people would often do if you're comparing, for example, like CO1, you would take the third base pair position, which mutates much faster than the other two, third base pair position in a codon, because it's degenerate very often, and you would just remove it. And then you would recreate your alignment from just the first two base pair positions that are under more constraint, right? Because changing them tends to change the amino acid. And so this was one way of dealing with the issue of back mutations, is actually pruning down the data set and removing loci with evidence of saturation. So either way, again, this is kind of a aside, but mutational hotspots, we know they exist, and they're easily modeled and accounted for. Okay, so now, assuming we've accounted for them, we should be able to evaluate a model of common design versus common descent by examining shared patterns of variation from regions of the genome not under sequence constraint. Importantly, these regions may or may not be functional. As an example, the internally transcribed spacer, ITS region of many ribosomal genes, is an intron that allows for alternative splicing. It has highly specific spliceosomal binding sites, but otherwise it shows no evidence of sequence constraint. It just needs to be present, right? It just needs to be there separating the two exons. Such regions are not expected to show any correlation between taxonomy and genetic identity because their specific nucleotide arrangement does not contribute to design, okay? And they're not expected to show correlations under a model of common design, I should say. So even if these sequences were originally created identically, let's say every single living organism, the creator just put the exact same sequence, just the exact same ITS region in the original pair, such that in the beginning there was perfect correlation between all organisms, independent mutational processes should have degraded any correlation relatively quickly. So in short, common design predicts a correlation only in regions under selective constraint for nucleotide identity. All right. And of course, we find nested patterns, nested hierarchical patterns in all regions of the genome, right? Including those with no evidence of selective constraint. I give a couple of papers. My God, you could go on, right? Just Google phylogenetics, look at all the results, and you can find thousands of papers that show this exact thing. And then I gave a personal example. I found similar patterns. I built some trees using what's called the loop region of RNA genes. So RNA genes have a 3D structure in which there's a loop and a pin or a stem. So the stem is like a folded part of the RNA gene or actually of the protein that needs to be bound together, but it creates this sort of loop above where there is no binding affinity up there, and that loop region just has high mutation. It's like it just mutates freely because it doesn't matter. They don't have to bind to anything. The stem tends to be very conserved because it needs to bind to its pair, right? And using these loop regions, I found nested hierarchical patterns of relationships. And in fact, the same nested hierarchical patterns that were recapitulated from protein coding genes. So they showed the exact same relationships, whether I used protein coding or whether I used regions under no sequence constraint. Didn't matter. Again, these patterns make sense given that DNA is the heritable material. It's the heritable material. It's like the thing that boggles my mind the most about creationism and about the idea of common design is that DNA is somehow not always the heritable material. That's just weird to me. It's a failure, and perhaps as I tried to elucidate a little here, maybe it's a failure on us as educators that so much of the general public views DNA as a language system, cells as machines, and biology writ large as mechanistic. We use silly words. We use words like machines, like the powerhouse of the cell. We use these kinds of engineering descriptors in textbooks because we think they're simplistic and help students understand. But I actually think they detract from reality and they make students have this sort of false view about what cells are and how they work. And then I say, instead we should be promoting the true view that DNA is a molecule, right? It's a nucleic acid. It's an acid. And it contains the same kind of quote unquote information as acetic acid or any polysaccharide or any other molecule, right? The information content is just other parts of the molecular structure. Like that's all that it is, right? It's an organic molecule. Understanding this, we can make sense of the messiness of genomic processes, right? Again, going back to elementary organic chemistry and understanding canonical and non-canonical products and the probabilities of receiving canonical and non-canonical products. Genomes are, first and foremost, the heritable material. They can design organisms all they want, but if they aren't passed on, they are no more useful than a protein in isolation. Okay, so TLDR, if genomes are 100% functional and genetic similarity reflects common design, then phenotypic similarity should always scale 1-to-1 with genetic similarity. Right? It must scale 1-to-1. Like I cannot understand how you would ever see two organisms that don't show that pattern when they're phenotypically similar, right? This is not observed in nature. As I gave a couple of specific examples above, you don't see this in nature. If organisms were created separately, genomic regions with no selective constraint for sequence identity should not form nested hierarchies, but they do. All right, so in conclusion, I hope I have adequately addressed your points in an informative way and that I have not mischaracterized your arguments, yada yada. I apologize for the length of the email. As you can see from the length of this video, this was a very long email and might, be contributing to the reason he hasn't responded. But my goal was to address as thoroughly as possible, like with detail and in-depth reasoning and studies. Like I tried to bring in as much as possible the arguments. No, I tried to not be hand wavy at all. Like give the actual data. But the problem, right, with doing that is it means that it's this stupid long thing. Like this is the thing that kind of frustrates me in these sorts of debates, right? Or when I watch these debates, it's like, how can you adequately address the misunderstandings of your opponent? If you think about evolutionary biology as a course in an undergraduate university, right? And any typical undergraduate university. At mine, it was required for you to have 2 semesters of intro bio, 2 semesters of chemistry, and a semester of genetics before you were allowed to take evolutionary biology. Do you understand what I'm saying here? You needed to be a junior, or at least like a second semester sophomore, to be able to take a course in evolutionary biology because there were so many prerequisites. There was so much basic biology that you needed to understand to be able to like get this course, right? You don't, in an hour debate, you don't have time to go through that. And so, and like, even this is, even as lengthy as this email is, I'm just glossing over so much. There's so much nuance and detail that I can't get into. Or I would be literally writing him a textbook. And it's unfortunate, but that's the nature of the beast. And it's the reason why, if they really want to understand evolution, they got to take some college classes. They have to take classes, right? They can't just sit around and debate atheists on YouTube. Like, you're never gonna learn biology that way. Nobody ever learned biology by atheist YouTube. Like, it's just not gonna happen. You're gonna always misunderstand it, um, and that's the nature of the beast. I tried to give a bit of a personal take here, that I'm not going to go into because it's not really relevant to this video, but I did try to give a little bit of a personal take that I hoped made him feel like I wasn't trying to attack him or his religion, whatever. But that's the thing that I did. I do want to read the PS. I had a postscript that I was thinking about that I thought I could do really quickly, so I'll go into that. And this is the concept of created heterozygosity. He only mentioned this very briefly, so that's why I didn't like dedicate a whole section to it. But here's the final paragraph. This is a curious notion that can be put to rest by simple Mendelian genetics. And so again, I make a mathematical argument here. So consider the first two humans were completely heterozygous at every site in the genome. Let's ignore the issue that chromosomes would be completely incapable of binding due to the lack of homology during meiosis. You know, a not a not little problem, right? If we assume complete linkage equilibrium to maximize the amount of diversity, that is, there's no linked sites, they're all, completely segregating independently by according to Mendel's law, we can predict heterozygosity into the future as follows. Noting from our discussion that the rate of drift is related to the population size, 2 because they're diploids, then the rate of loss of heterozygosity is the expectation of one generation after the initial, that's h sub 1, equals 2p, 1 minus p, 1 minus 1 over 2n. So this term, 2p, 1 minus p, you might recognize as the Hardy-Weinberg proportion for the number of heterozygotes, or the frequency of heterozygotes in the population. That's what that term is. And then this final term, 1 minus 1 over 2n, is drift. That's just the rate at which an allele is lost by chance. Okay, so the first term, again, is expected heterozygosity. Since n equals 2, they're Adam and Eve, 2 people, and assuming perfect heterozygosity, which is 0.5, right? That's exactly half and half. After one generation, h sub 1 equals 0.375. Just plug in the numbers, right? It's 0.375. That's a 25% loss of variation in one generation. Now, we can extrapolate this into the next generation, so the second generation of humans on the planet. But to do this, we must consider inbreeding, given that all subsequent mating is, by definition, inbred from Adam and Eve. So if we assume sib mating, so not mating back with a parent, but mating with siblings, this is a conservative measure. If you mate back with the parents, this measure is actually worse. But this is just assuming sib mating, which has an inbreeding coefficient of 0.25. After one generation, heterozygosity is then the Hardy-Weinberg proportion for heterozygosity, which is the 0.375 from our previous generation, multiplied by the inbreeding coefficient, gives us a heterozygosity in the second generation as 0.094. Thus, in only 2 generations, we have lost greater than 80% of the heterozygosity we started with. This is assuming that there's no mutations. So you can throw some mutations to try to bring it back, but given that we've only had 2 generations and the mutation rate, the empirically estimated mutation rate is like 10 to the negative 9, you're only going to get a couple of mutations, right? It's not going to be very high. And given that the population size is very small, the chance that those mutations are on chromosomes, especially again if we're assuming complete linkage equilibrium, the chance that they get passed on is incredibly small. So yeah, you're going to have very, very low levels of heterozygosity. So the concept of created heterozygosity falls flat immediately just based on basic Mendelian genetics. Okay, so all of that was really long. I want to close just by saying, if you're interested in the the religious component, like if you're interested in Bible translations or in Protestantism versus Catholicism, or what's the best exegesis for James 2. The book of James is a very fascinating book in the Bible, that in the New Testament, that has lots to say about salvation. So if you're interested in those sorts of topics, I recommend Standing for Truth's channel. If you're interested in learning about evolution, I don't recommend it. So that's what I want to close with. Thanks for sticking around. I know this was a really long video, and this was a really long email again. But hopefully you learned something interesting. And if you have any questions about any of the points that I made, drop them in the comments. I'll be happy to answer them. Hopefully coming up, looking forward, I would like to make some videos on the neutral theory of molecular evolution, specifically starting with sort of the history and the predictions and how they've held up into the modern age. That I think some people might find interesting. There's been, chatter about neutral evolution recently on YouTube, so maybe it would be a topic of interest for folks. So again, thanks so much for being here, and have a wonderful day.